Hi there. In this video, I'm going to be talking about the ribosomal RNA, or RNA, and ribosomes, and ribosomal RNA genes that are abbreviated rDNA. Ribosomes started evolving around 3.5 to 4 billion years ago, and they've expanded greatly since then, mostly in the initial first billion years of life. Anyway, there are differences between bacterial, archaeal, and eukaryotic ribosomal RNA genes. In bacteria and archaea, there's one long transcript consisting of a promoter region, a 5' external transcribed region, often abbreviated ETS for external transcribed spacer. And then there's a small subunit gene, usually around 16S Svedberg units. And then an internal transcribe spacer, and often there will be tRNA genes among or within the ITS. Then there's a large subunit gene, which is around 18 to 20 something S Svedberg units. So the Svedberg units relate to density in a cesium chloride gradient, and that's related to the length or size of that particular RNA eventually. And then at the end, there's a 5S gene often, which also becomes incorporated into the large subunit of the ribosome. Eukaryotic RNA genes are a little longer, so it's also in one transcript. So there's a promoter, a 5' ETS, and a small subunit gene, which is usually around 18S, but can vary a little bit. And then there are two internal transcribe spacers, ITS1 and ITS2, surrounding the 5.8S gene. And the origin of this is there was originally one spacer in ITS, and then so it has a certain secondary structure. ITS2 looks more like an intron that popped into the large subunit gene. So this end of the large subunit gene in bacteria is the same region as the 5.8S ribosomal RNA gene in eukaryotes. And there's this ITS, which looks something like a group 1 intron that's popped into that space. And then there's a large subunit gene, usually around 25 to 28S, and then a small 3' external transcribe region. So these 3' ETSs are similar in both of these. The 5S gene in eukaryotes can be either in this location, or they can be on different chromosomes at a distant location. And remember that the ribosomal RNA genes are transcribed by RNA pol 1, and the 5S genes are transcribed by RNA pol 3. So they are transcribed, that 5S gene can actually be in either direction. Here's another view of those two types of genes. There's the bacterial, there's the eukaryotic, and showing which parts end up in which subunit of the ribosome. So the small subunit is the primary functional unit of the small ribosomal subunit, while the large ribosomal subunit is composed of the large rRNA and the 5S rRNA, as well as in eukaryotes the 5.8S ribosomal RNA. And it's the large ribosomal subunit that has the peptidyl transferase, although both subunits have sites that will attach the tRNAs. The main function of the small ribosomal subunit up here is to deliver the mRNA to the large subunit, and also it performs the ratcheting, moving the mRNA through the ribosome 3 nucleotides at a time during translation. Because ribosomes are required in such high numbers, from about 10 to 100,000 ribosomes in a bacterial cell and between 1 and 10 million in most eukaryotic cells, although there are some that are even larger. The rRNA genes are transcribed almost continuously through the life cycle, such that in a cell, Roughly 80% of the RNA in a cell is made up of ribosomal RNA. And they do this by loading up the gene with RNA polymerase to transcribing here and then moving all the way to the end where you've got a complete transcript. So they load up approximately 100 RNA polymerase molecules on the gene. So the basic rate of transcription is about 50 nucleotides per second, and if you multiply that by 100, then for each transcript you're getting approximately 5,000 nucleotides per second. This still isn't quite fast enough to meet with the life cycles of those cells or the replication cycles, so they actually have to have multiple copies of these as well, most cells anyway. For example, E. coli has about 7 copies, or most E. coli have 7 copies. This is enough so that it can replicate or divide every 20 minutes, 20 to 40 minutes. Humans have, on average, about 200 copies per haploid genome, so 400 copies per diploid, and that's just enough to make a couple million ribosomes in about a 24-hour period. Upstream from the promoter region, there are these repetitive areas that actually bind transcription factors and RNA polymerase so that they're ready to pop onto the real promoter to start transcription of the genes. Often people have called this region here the non-transcribed spacer, but it's actually, you see these little short pieces here. Those are actually short pieces of RNA, and they actually have a purpose to control transcription of this gene. But so this region actually does transcribe RNA to a certain degree. In bacteria, usually there are multiple copies, although not always. And they're dispersed. So in E. coli, it's 7 copies. There are a few bacteria with only 1 copy of the ribosomal genes, and they divide very slowly. So the more copies you have, the more ribosomal RNA you can produce in general, and the more ribosomes you can produce, so that you can have your cells divide more often, because then you can have enough ribosomes for both of the daughter cells. So here's one with 5 copies, as I said. So that would probably mean that this one could divide every maybe 30 to 40 minutes, whereas if you have 7 copies, you can be like E. coli and divide every 20 minutes, 20 to 30 minutes. Ribosomal RNAs are transcribed as a long transcript consisting of several parts, in this case the small subunit, the large subunit. In bacteria and archaea, this would be the small subunit, large subunit, and often the 5S gene as well. So there's one long transcript here, and then there's processing, and this processing is done by RNAs primarily, not by proteins. And it's not splicing, so it's actually processing. The pieces are cut apart and not spliced together. And then this small subunit becomes incorporated into the small ribosomal subunit, and the large subunit and the 5S become incorporated into the large ribosomal subunit. So looking at this in the electron microscope, in eukaryotes you have tandem repeats of these transcription subunits, and each one of these looks like this with about 100 polymerase molecules on it, and then each one of these at the end becomes a transcript here and then processed in this case into the 18S, 5.8S, and 28S, or sometimes 25S depending on the organism. And the 5S genes are transcribed elsewhere and then incorporated into the large subunit along with the 28S and 5.8S, and the 18S becomes the major RNA in the small ribosomal subunit. Rather than being dispersed, in eukaryotes, The ribosomal genes are located in a locus with multiple copies, multiple tandem copies. And there can be, depending on the species, more than one of these loci. In this one, which is from broadbean, there's only one locus on chromosome number 1. In peas, there are 2 loci, and in some there are more than 2 loci. But they're all in large tandem repeats. In this case, for Vishafaba or broadbean, there's an average of 250 copies of these, one after the other after another. Although some of the cells have up to 23,000 copies, all in tandem. Each one of those transcription units looks like this. So there's the small subunit, the 5.8S gene, the large subunit, the 2 internal transcribed spacers, and then upstream in the intergenic spacer, IGS, there are these repetitive elements. And one part of the repetitive element looks like the promoter. So that binds transcription factors, which then bind the RNA polymerase, and then load them up on this site, as you saw in one of the previous slides. And depending on the intergenic spacer, there can be anywhere from 1 of these repeats up to 35 repeats, all in the same location. And in Vishafaba, these are 325 base pairs long, although they consist of a sort of 2 subrepeats of 155 base pairs each, and then some unique sequence. In most species, they are around 150 to 200 nucleotides each, or base pairs each, and they all are active in binding transcription factors and RNA polymerase to that site. Processing of the ribosomal RNA is a stepwise process. So here's the DNA up here. Here's the primary transcript. In this case, it's 35S, and this one's from yeast. It would be similar in humans, though, and similar in plants. All of this processing is done by other RNAs, these SNO RNAs, or small nucleolar organizing region RNAs. So the first thing that happens is that promoter region is cut off by the RNA, and then a further piece is cut off from there, and then there's a cut within ITS1, so now you have a 20S on this side, and a couple of different possibilities for this one, either a 27SB small piece or a 27SB long piece, so varying by a little bit of extra RNA kept on the 5' side of the 5.8S gene. Then there's further processing from there. The final processing of the 20S into the 18S is done, and that winds up in the small subunit. And then each of these 2 pieces is further processed by cutting the ITS2 out and then shortening that further into the 5.8S and 25S, or into the 5.8S long and the 25S. And those each wind up in the large ribosomal subunit. In the case of plants and fungi, this is a 25S fragment. In humans, it's a bit longer, being a 28S ribosomal RNA. In bacteria and archaea, both transcription and translation occur in the same compartments. So you have transcription by RNA polymerases up here for standard genes, most of the genes. And you also have transcription of the ribosomal RNA genes, assembly of the ribosomes, and translation by those ribosomes in the same compartment. In eukaryotes, these are split. So you have transcription of the standard genes here, as well as the ribosomal RNA genes. These come together. The ribosomes are assembled into the small subunits and the large ribosomal subunits. They're exported out through the nuclear pores, and the small subunit doesn't have much problem getting out, but the large subunit needs to squeeze through those nuclear pores because it's a very large protein RNA complex. And then you get translation of those mRNAs out in the cytosol. Some of those RNAs are for ribosomal RNA proteins, as well as other things like RNA polymerase. Those are shipped back into the nucleus to function there and to form the ribosomes for translation. So transcription of all genes in eukaryotes is done in the nucleus, except those in the mitochondria and plastids. But the translation occurs out in the cytosol, and then some of those proteins are shipped back into the nucleus, where assembly of ribosomes takes place, which are then shipped back out into the cytosol to do translation. So transcription of the main ribosomal RNA and assembly of the ribosomes occurs in the nucleolus, within the nucleus. And the nucleolus is an area of the nucleus, and it's not a membrane-bound organelle. So what you find is there's a fibrillar portion, which is where you're getting the ribosomal RNAs transcribed. And then there's a granular portion where you start to see the proteins, the ribosomal proteins, assembling around that ribosomal RNA to form the ribosomes. And then they're eventually shipped out into the cytosol, where they find an mRNA, which has been transcribed elsewhere in the nucleus. And some of these then translate into ribosomal proteins, and those are shipped back into the nucleus and into the nucleolus for that process of ribosome assembly. So here are a couple of electron micrographs of that, of the nucleolus. Here's a cell here, and then there's the nucleus with heterochromatin and euchromatin, and then this nucleolus. And if you look very closely at the nucleolus, you find what's called a fibrillar portion, which looks a little lighter. And if you look really closely, you can see little strands, probably of RNA and DNA going through there. And then a granular portion where the ribosomes are assembling in that region. Just to remind you of where this may have come from, all of this, the nucleus and the nucleolus, this is a electron micrograph of a planktomycete, a bacterium, that has this double membrane, or actually single membrane, folded over on itself. That looks like a nucleus with ribosomes inside. And it has an area which contains chromatin, which is called the nucleoid, mainly the chromosome. But it also has different areas within there that may represent places where ribosomes are being assembled. And again, this is one possibility, this organism or organisms like this, that were the sources of a nucleus as well as internal membrane-bound organelles in eukaryotes. If you build models of the ribosomal RNA, this is the small subunit, you find some regions are double-stranded, other regions form bubbles and loops, internal and external loops, that is. So this is one model for E. coli. The 5' end is right here, and then you go through different domains, this domain up here, and then through this domain, and finally this final domain down here with the 5' or the 3' end being here, ending in what's called the anti-Shine-Delgarno sequence there, and it attaches to or hybridizes to the Shine-Delgarno sequence of the mRNA in bacteria. So that's the first region that hybridizes to the ribosomal RNA and the ribosomal ribosome small subunit, which attaches that RNA to that ribosomal subunit. So here are those different domains. Here's the first domain in red, and then the second domain in green, and then the third domain in yellow, and then this fourth domain with the anti-Shine-Delgarno sequence. And you can see in the ribosome, when you put them all together, that these do form definite regions of that small subunit. And these are the large subunits that will connect up with the small subunit ribosomal RNAs, or the small subunit ribosomes. And again, it's the small ribosome subunit that first attaches to the mRNA and then delivers that to the large subunit, and then has this ratcheting function to move that mRNA 3 nucleotides at a time. And it also has the sites for the tRNAs to attach as well. And here's what the large RNA subunit looks like, starting from domain 1, moving up to domain 2, 3, 4, 5, and 6. It's this region in domain 5, this circular region right here, is the peptidyl transferase, the enzymatic portion. Other portions, such as this, and this, hold the tRNAs, and then other regions are for attaching to the small ribosomal subunit. As with all RNAs, including the large subunit, the small subunit, and virtually any RNA, you will have these loop-out regions, as well as some double-stranded regions, but the double strands don't go for any extended lengths. Usually it's 7 base pairs at the most, and then they kind of fall apart into a loop, these loops can interact with loops over here, and these internal loops can interact with other loops on the other side as well. So here are a few other models of the ribosome and ribosomal RNAs. Again, there's the small subunit. The anti-Shine-Dalgarno sequence would be right there. The 5' end is there, so this is kind of flipped around with respect to the first one I showed. And then here's the small ribosomal subunit there, the large ribosomal subunit there. There's the tRNA in the A site, the tRNA in the P site, and then the exit site would be over here. The mRNA is threaded through here, so the peptidyl transferase of the large subunit would be somewhere right in here, in that area. Here's just a ribbon structure of the small subunit, and then here are the large and small subunits pulled apart, kind of like opening up a cookie. So you can see how they meet up with the A, P, and E sites, and here it's a reverse because you're looking at this as it's unfolded, essentially. So that's the A site bringing the amino acid in, there's the P site holding onto the polypeptide, and then there's the tRNA in the E site that has released its amino acid already. And this is the reverse over here. So the mRNA would be threaded through up here, and the polypeptide would be growing down here and threaded through a tunnel through the large subunit. Here are some higher resolution views of that, and this is just showing the ribosomal RNA, not showing all the proteins. But this is a top view, so you see the small subunit from the top, small, large subunits from the side. So the darker color is the small subunit, there's the large subunit, and there's one of the tRNAs that you can see there. And then this is from the bottom, looking at the large subunit, and then this is from the other angle, looking at the large subunit here, the small subunit there, and the tRNA in between, being held essentially by both the large and the small subunits. Here's another high-resolution view, but this is with the large subunit and the small subunit pulled apart, so you can see the tRNAs in the A, P, and E sites of the ribosome for both the small subunit over here and the large subunit over here. This one would be the tRNA holding the amino acid. This would be the tRNA holding the polypeptide, and then this one here would be the tRNA without the amino acid that's on its way out of the ribosome. The peptidyl transferase site would be right there, and the tunnel would be adjacent to that site. Here's a schematic view of that, showing the mRNA being threaded through, attaching to the tRNAs at the A and the P sites, and then transferring the amino acid to the growing polypeptide chain over here, which is threaded through a tunnel, and then these shift over, ejecting the tRNA at the E site and then dislodging the polypeptide chain from that one in the P site, moving that over to the E site, moving the one in the A site over to the P site, which now contains the growing polypeptide chain, and then bringing in the next amino acid into the process of translation. Just to show you another part of this process, so this is a fairly high resolution shot of just the ribosomal RNAs with a few proteins added on here. But here's the tRNA at the exit site, there's the tRNA at the P site, there's the one at the just coming into the A site. So the peptidyl transferase function would be here, and then the mRNA would be threaded through the ribosome here. But there's also another thing involved over here, and that's a protein, which is an elongation factor for translation. And this one's called EFTU. And this one acts as a check on whether that new tRNA entering that site is being held there very well by the hydrogen bonding between the mRNA codon and the tRNA anticodon. So as the tRNA enters that A site, this EFTU out here will then clamp down on it, and if it's held tightly, it will allow translation to continue. If it's not held tightly, it'll open up again and it will eject the tRNA and allow another tRNA in there to check whether it's bound well to the anticodon. And EFTU is just one of many elongation factors, and I'll be talking about that in a few minutes with regard to initiation factors, elongation factors, and release factors in translation. Ribosomes vary in size depending on the evolution of those organisms. So the simplest ribosomes of 4 billion years ago were even simpler than these. But in bacteria, these ribosomes evolved approximately 3 to 3.5 billion years ago. And they're of a certain size, in this case 50S and 30S, although that varies somewhat depending on the type of bacterium. And then in eukaryotes, they're quite a bit larger. They're 60S and 40S subunits for the large and small subunits. And archaea are slightly larger than those in bacteria. And then if you look at the ribosomes in organelles such as the mitochondrion and the chloroplast, they're a bit smaller because they started out as bacterial ribosomes, but they've lost a little bit in terms of size and abilities because they only translate to a limited extent in those organelles. Here are a couple of other views of those for bacteria on the left and eukaryotes on the right side. And if you look up here, here's the small unit, subunit, small ribosomal subunit from the top, and then from the inside, and you can see the A, P, and E sites, and the large subunit down here from essentially the bottom, if you look at it that way, and then the inside, the A, P, and E sites there. You can see that the small subunits are a little smaller in bacteria than eukaryotes, and they have fewer sort of side regions that actually interact with other proteins. So these central inside pockets have remained fairly constant for about 3.5 billion years or more. But then these regions on the outside where they're contacting other proteins and parts of the cell, those regions are more prone to change. And the same for the large subunit. These interior portions are fairly well conserved, but the outside regions that are now interacting with other proteins on the outside and membranes and things like that, those have changed more rapidly during evolution. And for example, you can see this little arm on this large subunit of the ribosome that attaches to specific proteins during translation. And then this is an added arm in eukaryotes that you can see that attaches to proteins. And there are other examples of those regions on the outside fringes of the ribosome that are interacting with other things that affect translation in those different organisms. Here is a diagram of the process of translation, including an initiation phase, an elongation phase, and then a release phase. So if we start over here with initiation, it starts with the small subunit and then an initiator tRNA, which is the AUG for methionine. There are also some initiation factors that come together to join that with the small subunit of the ribosome. Then this joins with the large subunit, and the initiating factors come off, and then a tRNA with the EFTU, which is an elongation factor, come in and then initiate translation. At this point, the EFTU, the elongation factor TU, positions the tRNA with the amino acid at the A site. It then tests whether the bonding between the anticodon of the tRNA and the codon of the mRNA is firm. If it is, it leaves it in that position and then pops back out. And then the elongation factor G, which is a G binding protein, GTP hydrolysis, which causes a shift in the mRNA with regard to the ribosome by 3 nucleotides or a triplet codon. Release of EFG, and then another tRNA comes in, and you go around and around the circle until a stop codon is reached. And this then causes binding of a release factor in that cleft where the next amino acid should come in. This causes a hydrolysis of the nascent peptide, so that comes off, the polypeptide comes off, and then some of the release factors are dissociated from the ribosome, and some other release factors pop in to pull apart the large and small subunits of the ribosome, and they then cause release of the mRNA, and then you start the whole process over again. Here is a comparison between bacterial translation and eukaryotic translation. So the slide before was on bacterial translation. And so you can see here it is on this side with the initiation factors and then the elongation factors and finally the release factors here. And some of the same things occur over here on the eukaryotic side, but there are just more of them. So there are more initiation factors. There are more elongation factors, although some of these are analogous to the elongation factors in bacteria, so they haven't completely changed. And then there are a number of extra release factors as well. One thing to notice here also during initiation, there's a Kozak sequence which causes circularization of the mRNA. So it is recognized by the cap binding protein and then other initiation factors, and then those join with the small subunit of the ribosome in the cytosol. And next, after initiation, you get release of those initiation factors and then addition of the elongation factors. And again, this can happen over and over and over again to add more and more amino acids to the nascent protein that's coming out of the large subunit. And then finally you get to the end, a termination signal that is a stop codon, and that initiates release factors to come in and pull apart the two subunits, the large and small subunit, and then release the mRNA and start all over again. So that's a short version of ribosomal RNA and ribosomes, as well as translation, which is accomplished by the ribosomes. Again, these are macromolecular structures consisting of several rRNAs and dozens of proteins, and the proteins are primarily there to hold the RNA into the functional form of the RNA, including the peptidyl transferase, the enzymatic portion of the large subunit. Okay, that's all about ribosomal RNA genes, and next time I will talk about tRNA genes, mRNA genes, and other small RNA genes as well. So thanks for watching and listening, and I will talk to you later.